Let's talk a little bit about the evolutionary developmental biology of regeneration. And in this case, I want to start by just describing a few different ways that different animals regenerate. One you might be familiar with is autotomy, which is where a part of an organism can be lost, say the tail of a lizard or a skink is broken off, and then that organism can regenerate that part of their body that's missing. And in animals, for example, in humans, we are very limited in the types of regeneration that we can perform. We can regenerate some tissues, but in general, we can regenerate the tips of fingers, for example, if they're cut off, but that's about it. We can't lose an entire limb, for example, like some other organisms, even vertebrates like axolotls. So it's really of interest for evolutionary developmental biologists to understand how organisms can regenerate tissues. There are a few different basic categories of regeneration. One is called paratomy, which is where two organisms grow in the same body and then they split. So in this case, we're looking at two different pictures of worms, nematodes, that undergo regeneration or replication by paratomic fission, which is, here is the head of one organism that's growing essentially on the tail or the posterior end of the original organism. So we can see a head developing and then all the different body parts. And eventually this organism will undergo fission or split. The tissues will separate and produce two different organisms from the original. So that's a type of regeneration, paratomic fission. There's also paratomic budding, where an individual can replicate its tissues and in fact other organisms by having a small section of tissue start to generate a new organism from the parent. So in this case, this is really interesting. The parent tissue is oriented from the anterior to the posterior here, and the bud adopts the opposite orientation, where the anterior is the part that grows out from the parent tissue and eventually creates a second organism. Another form of budding involves a separation down the midline of the organism, where the anteriors and the posteriors are here, and growth eventually leads to budding of one organism from the second organism. Planaria are some of the best studied species in terms of regeneration, just because historically, they were essentially fun to play with by older developmental biologists. So in planaria, these basically indicate evidence of archotomy, which is the splitting of two organisms. So if you cut an organism and it creates two heads, for example, eventually those organisms will divide down their somatic tissue and produce two different organisms that basically create on the left side the right side of the body that's missing, and the organism that's forming from the right side of the tissue will generate the left side of the tissue that's missing. And a number of groups, including Alexa Bailey's lab, have indicated, using these sorts of phylogenetic trees, they've reconstructed the evolutionary developmental biology of regeneration. The idea here is that if you look across huge groups of organisms, in this case, we're chordates, so we're here in the deuterostomia, so humans are part of the chordates. And in our group of organisms, there are organisms that can undergo whole body regeneration and also structural regeneration. And in this case, that would be, like I indicated before, axolotls that are vertebrates that are able to undergo all sorts of different types of regeneration. For example, limbs, which is structural regeneration. So if you look across the tree of life, it's really interesting to see that there are lots of different groups of organisms that can undergo various types of regeneration. But in some types, those types of regeneration have been lost. So it's really interesting for evolutionary developmental biologists and evolutionary biologists in general to ask the question, if regeneration is such a strong benefit, or at least we think it's a benefit, what would cause it to be lost? And why isn't regeneration an ability of all sorts of organisms? Because the ancestral organism to all of these species, based on the outgroups, porifera, and then some of these more lately diverging, or earlier diverging clades, Placozoa, Ctenophora, and Cnidaria, they all have the ability to regenerate. So why is it that what we colloquially refer to as the advanced species, like chordates and vertebrates, why is it that humans have lost the ability to regenerate? So it's of interest, again, to understand the genetic mechanisms that underlie the ability of organisms to regenerate either tissues or entire bodies. And back to planaria. One of the most famous scientists to study planaria over a century ago was Morgan, who reported that if you dissected planaria, sometimes they wouldn't always regenerate the missing part. And he found in particular that if you took a very small, if you sliced across the body of a planaria, and you've got a very small piece of tissue, that it would regenerate two heads, instead of a head on the anterior and the tail on the posterior. And so he called this phenotype Janus heads, based on the Roman Janus, which was an organism deity that had two heads, a god that had two heads. So he termed this phenotype Janus heads. And he found that there were a number of different ways you could physically cut a planarian, that instead of allowing it to regenerate normally, which is to produce both a head and a tail, that it would produce two heads. So that is essentially what we're going to be looking at in terms of regeneration, is why is it that some types of amputations of planaria don't allow them to regenerate properly? And what at the cellular and the molecular level causes the ability in some cases of planaria to regenerate properly, and in other cases not. Before we get into details, it's important to know some about the anatomy of planaria. So planaria have photoreceptors. They have cells that can detect light. They're very primitive eyes. They're not eyes the way we think of eyes in mammals. But they do have photoreceptors. And so we call these eye spots, and that's an indication of the dorsal and the anterior end, of the planaria. So this is a dorsal view. It's looking sort of at the top of the organism. If you flip it upside down and look at the ventral surface, you see a pharyngeal opening, which is really cool, but we don't have time to get into how exactly planaria eat, but they basically extrude their stomach out of the body and use it to engulf food and then pull it back inside the body. It's really cool looking if you've never seen it before. And then a gonopore related to mating. So that's how we can tell the anterior of a planaria. Basically, we want to look at where those eye spots are located. They look sort of cross-eyed because the pigmented photoreceptors are in the middle, sort of towards the midline of the body. So planaria do look sort of cross-eyed, but that's how we tell where the anterior of the planaria is located. This is another diagram that just shows a number of different ways that people have cut planaria to produce regenerate. So for example, in figure 5 here, if you cut a planarian, an adult planarian, down its midline, it will regenerate both the left and the right sides to produce two, essentially, offspring that are complete planaria. But if you do something like cut off the head and then make a short incision down the midline, you can generate two different heads and a regenerate from the head itself. So the head will regenerate a tail, but somehow the body of the planaria, the posterior, gets confused. And so if you make that vertical here incision, it will grow two heads instead of one. So all of these different experiments producing all sorts of sort of weird planarial regenerants, again, poses this question of what is happening at the molecular level inside the organism that's directing the cells to adopt specific fates, and to know, for example, how many heads a section of the tissue of this planaria should produce. When a planaria is truncated, let's say they cut off the head of the organism, one of the first things that happens is that stem cells will migrate to the cut site. And those are unpigmented cells, so they're very easy to see in a planarian. And we can see here the regeneration of eye spots in this organism had its head cut off. And it's those migrating stem cells that form new tissues that are of particular interest. How do those stem cells, once they arrive at the position where an amputation occurred, how do they know what types of cells they're supposed to produce as they divide and differentiate into mature somatic tissues? What sort of signals are they getting from the body of the organism that tells them, hey, you're sort of towards the anterior of the body, so you stem cells, when you divide and produce daughter cells, as you differentiate, you should turn into head cells or head-like cells instead of tail cells or left side cells or right side cells. So what we're going to look at is all about cellular signaling, how cells know where they are in the context of the whole body of the organism. And another question that we'll ask in general is, why did these sorts of amputations produce Janus heads phenotypes? What about this section of tissue, when it regenerated, why was it not able to produce a head on one end and a tail on the other? So what sorts of parameters define the ability of these sorts of organisms like planaria to properly regenerate versus not? So it's possible that things like the dimensions, maybe the height versus width. So maybe if you make a short fragment of tissue that's relative to its width very short, maybe that somehow disrupts regeneration. Or maybe it's temporal. Maybe if you make a very quick cut and wait for a generation time that's very long, there's some sort of ability of the regenerative ability to get confused. In other words, maybe if you make the anterior cut early, but then you wait a long time to make that posterior cut, maybe that produces a section of tissue that's able to regenerate properly. But maybe if you cut the anterior and the posterior sections very soon after each other, one after the other, maybe that sort of confuses the body. But again, we're trying to figure out what is happening at the molecular level that allows regeneration to proceed as it normally does in planaria. So early on, it was clear that what has to be happening for a cell to know, especially a stem cell that has migrated to the site of an amputation, there has to be some sort of communication between cells, with cells that are at the site of amputation communicating to the cells nearby, the stem cells that have just arrived, telling them, giving them some sort of information about where they are in the body. Are they on the left side of the body or the right side of the body? Are they dorsal or ventral? Are they anterior or posterior? So it seems like each cell in the body, or at least most cells in the body, must have some sort of message that they send, or some information that they have, that tells the cells around them, hey, we're anterior dorsal cells. So next, let's take a look at the sorts of information we already know about how cells communicate with each other. There are 4 different major groups of signaling systems that cells use to communicate with each other during development. We have juxtacrine signaling, which is sent from one cell physically to its neighboring cell. So these tend to include molecules that are present on the cell surfaces of cells that are physically touching each other. So these juxtacrine signals are very short range. from one cell to the immediately adjacent cells around them. There are paracrine signals, which are sort of hormonal type signals where a chemical or a molecule is released from a cell and it is sent into the immediate vicinity. So not just the physically neighboring cells, but maybe cells 2 or 3 or 4 cells away. And that's paracrine signal. It involves release of some sort of chemical or molecular signal that diffuses over a short range. That's compared to the endocrine signaling pathways, which distribute throughout the body. So in particular, in example, humans, those endocrine hormones might get into the bloodstream and therefore be distributed throughout the entire body of an organism. And then sort of a fourth category, which is also sort of a subset of paracrine signaling, is autocrine signaling, which is the idea that paracrine signals, these short-range molecular signals, can be detected not only by the tissues, the cells that are immediately around the signaling cell, but that signaling cell might itself stimulate itself by releasing the chemical and then recognizing that it released the chemical and responding accordingly. So that's auto or self-crine signaling. And in particular, let's look right now at two different paracrine signaling pathways, the Hedgehog pathway and the Wnt pathway. The Hedgehog pathway was initially discovered in Drosophila, involves a particular ligand or a protein that is bound by a receptor protein. The ligand itself is called Hedgehog, abbreviated HH. And it has one copy or one gene in Drosophila, fruit flies, called Hedgehog. And when that gene is mutated, instead of causing these strips of gene expression, this is gene expression of the Hedgehog gene, the production of the Hedgehog protein, which produces these denticles or short sort of hairs in strips on the developing Drosophila larvae. When that Hedgehog gene is mutated or lost, instead of organizing the expression of Hedgehog into strips, which is its main job, which is Hedgehog is involved in determining the anterior, posterior, or in this figure, left, right, orientation of cells within each segment of the arthropod body. If you delete Hedgehog, all of those segments get confused about which end of each segment is anterior and which end is posterior. Anterior on the left and posterior on the right in this example. So in the Hedgehog mutants, those denticles, those short bristles or hairs, are found across the body of the organism, making it look like a hedgehog, which is why they named this mutant the Hedgehog mutant. And there are multiple different orthologs, or paralogs rather, of the Hedgehog gene in vertebrates, and those are called, for example, Indian hedgehog, sonic hedgehog, and desert hedgehog. So in short, Hedgehog is a protein that's produced by a gene that's one of these signaling proteins that can diffuse over short distances to signal from one cell to other cells. And this is how Hedgehog works inside the cell. On the left here, we have a diagram of the gene regulation cascade that starts with the Hedgehog protein and involves other proteins called patched smoothened CI, which stands for cubitus interruptus, and then ultimately the response, which is transcription. So as we've discussed, the idea between cells signaling to each other is the effect of the signal, one cell telling another cell some information. The output of that signal should be a change in gene transcription. The signaling cell intends, although it doesn't have a brain, to have other cells respond by changing the types of transcripts and thus proteins that they produce. And those sorts of effects could be things like they change the fate of cells, or they change the ways cells activate or are otherwise engaged in regeneration in this case. So in this particular example, in the Hedgehog pathway, there's a protein called pKa, which normally, in the absence of Hedgehog protein, causes the phosphorylation of this protein, cubitus interruptus. And when CI is phosphorylated, it gets cut in half, cleaved. And what happens then is part of the cubitus interruptus protein enters the nucleus and it binds to its transcription factor binding site. So CI is a transcription factor, but when pKa is present and active, only part of the CI protein, after it gets cleaved, enters the nucleus. And that is the transcription factor binding domain of CI, that part of the protein that binds the promoter region. But without the top, in this case, part of that protein, all that CI does is bind the promoter. And what that does is actually represses transcription of hedgehog responsive genes. So this happens not just in one location on one gene, but across the genome. Anywhere the CI transcription factor would normally bind, it will bind, but no transcription takes place because, again, that protein is missing its transcriptional activation domain, which is the top half of this protein as drawn. Now, it's important to note that there's a cell surface receptor called patched, and it is physically associated in the cell membrane with a protein called smoothened. And when patched is present in the absence of hedgehog, its physical structure binds to and prevents smoothened, a different protein, from acting. Now, on the right side here, what smoothened will normally do when it's active is it inhibits pKa. So here we go with the inhibition of pKa on the left side of the screen. When hedgehog binds to patched, that causes the structure of patched to change, the physical shape of the protein patched, so that it can no longer physically inhibit smoothened. And then what smoothened does is it represses pKa, which itself is a repressor of cubitus interruptus. So they're missing a step over here in this, which should be that smoothened, let's draw it out, inhibits pKa, which itself inhibits cubitus interruptus. So that double negative is why they show smoothened positively regulates cubitus interruptus. So in the presence of hedgehog, the repression of a repressor allows cubitus interruptus not to get phosphorylated, therefore not to get cut in half, and the entire protein enters the nucleus, where cubitus interruptus acts as a transcription factor that recruits other transcription factors and allows the transcription of what are called hedgehog response genes. And that's because when hedgehog is present, this pathway allows the transcription of genes. So those genes that are produced when hedgehog is present are therefore called hedgehog response genes. So that's your introduction to the hedgehog pathway. Now there's also another important pathway that we need to know about, and that's the Wnt pathway, which stands for wingless and integrated. And in this case, Wnt, this again, signaling molecule is passed from cell to cell, so it goes in between cells and signals to nearby cells in a paracrine fashion. Wnt is often involved in the posterior development or identity of organisms. So like hedgehog, which is a segment polarity gene, it tells segments of tissues where they are in the anterior to posterior dimension. Wnt is also one of these developmental proteins that's involved in posterior specification. So again, it may be likely involved in things like planaria knowing where their head is and where their tail is, and if they get amputated, where the relative head of the chunk of tissue is and where the relative tail of that piece of tissue is. And in this case, the key player here is a protein called GSK3, which is involved in destroying normally a transcription factor called beta-catenin. So GSK3 normally will cause degradation of this transcription factor, beta-catenin. But when Wnt is present, it binds on the outside of a cell surface receptor called frizzled. So Wnt activates frizzled, and when frizzled is activated, it activates this protein called disheveled, and disheveled inhibits GSK3. So when Wnt is present, GSK3 is turned off. That prevents the degradation of beta-catenin. So beta-catenin can now enter the nucleus and in association with left TCF proteins bind to DNA and cause transcription of Wnt-related proteins. So that's the Wnt signaling pathway in a nutshell. So that's the developmental genetics and molecular biology of some of the important gene regulatory cascades that are involved in polarity determination as related to regeneration. Now let's turn for a minute and look at a reagent, a technique, and a concept that are particularly relevant for understanding how these pathways work and how they regulate each other potentially. So the first of these is an EST library, which is a type of reagent. EST stands for an expressed sequence tag. The concept here is that if you take a cell, and if that cell has a bunch of different messenger RNA molecules, which I'm going to just indicate as different colors and different numbers of the different colors, it's often useful to know how many and which types of messenger RNA are being produced at one point in the cell. And that's important to understand how transcription factors influence the expression of genes. So an EST library basically means you break open the cell and you collect little fragments of each of these different genes that are being expressed, so transcripts. And so that would constitute what's termed an EST library. Again, expressed in this sense means messenger RNA. Sequence, I think, is obvious. It's we get the sequence of these different ESTs, which are, again, fragments of messenger RNAs. And that's what tags indicates. It's not the full messenger RNA sequence. It's a portion of this sequence. And it's enough sequence from each of the mRNAs to uniquely determine which specific gene it's from. So the big picture is this is a way, and was before whole genome sequencing and RNA-seq was something that we could actually do in the laboratory. This was 10 or 20 years ago, a way to sort of catalog for a specific cell which genes are being translated, or transcribed rather, and maybe translated in a particular cell at a particular time. And one of the things you can do with an EST library is perform quantitative reverse transcriptase PCR. And I'm not going to go into detail about PCR yet, although there are a number of good videos that you can watch that explain what PCR is. But in brief, PCR is used to make millions, literally billions of copies of a particular sequence of a chromosome, basically for the purpose of detection or sequencing. And in this case, what you can do is take your cDNA library, that is that EST library, and perform quantitative reverse transcriptase PCR on that library. That's what allows scientists to quantify, that is to count up, the levels of transcription of each gene that are being transcribed, each gene that is being transcribed by a particular cell at some point in time during development. So the way that works, again, in brief, is you take a messenger RNA molecule and you use the enzyme reverse transcriptase to convert that messenger RNA to a complementary DNA molecule. So reverse transcriptase is a protein that's often found in viruses that does reverse transcription. It takes an RNA molecule and produces DNA out of it. So you can get a double-stranded piece of DNA from an mRNA molecule, and that's what's essential for polymerase chain reaction. The two strands of that cDNA are denatured or melted, and then oligonucleotide primers base pair these are designed specifically to do this, base pair with those two template molecules and allow RNA polymerase to finish synthesizing those two pairs of template strands to produce two double helices. So you go from one double helix to two double helix, and then that polymerase chain reaction step is an exponential growth process where each of those templates is duplicated exponentially. And over the course of about 30 cycles or so, literally billions of molecules of DNA representing an original messenger RNA molecule that was present in the cell. Those copies of the messenger RNA, the PCR products, can then be quantitated by simply, although it's not simple, by simply figuring out how many molecules of this double helix are present inside the reaction as it proceeds. So again, in sum, cDNA libraries, or ESTs, are basically a collection of messenger RNA from a cell or from a tissue or from a whole organism. And it's predicted, it's anticipated, that the relative number of transcripts has something to do with the amount of transcription of each gene that's going on in that cell or tissue or organism. And the way that we can quantify or count how many messenger RNA molecules were in that tissue sample is by amplifying that single, or maybe a few, or maybe several molecules of messenger RNA to create literally billions of them through the process of PCR, and then counting how many molecules were produced after PCR. In terms of interpretation of quantitative RT-PCR data, there's a particular concept, RGE, which stands for relative gene expression. And this is a really critical concept to understand both how to perform quantitative RT-PCR analysis, but also how we, as bystanders and interpreters of qRT-PCR data, need to analyze these data. So this is really important. So here's the scenario. Let's hypothesize that we have a cell and it's got two conditions that we're going to experimentally vary. One is a control condition where we have a cell or a tissue or an organism and we do nothing to it. So that's the normal organism. And let's say we hypothesize that we have a particular experimental treatment, RNAi, knockdown of another gene, or changing the temperature or the pH of the organism, or growing it in more dense or less dense conditions, whatever it is. There's an experimental condition, and our hypothesis is that will cause the increase of expression or transcription of the gene that's coded in blue here. The squiggly lines then indicate two different types of transcripts from two different genes. The red gene, shown here, there are 4 different transcripts in this cell, 4 different messenger RNA, and that gene is a standard gene. And what that gene is scientists choose whatever that gene is to represent a gene that they don't expect, given what we know about biology and about the organism and about this experimental design. We don't expect that gene's transcription amount to change between the control treatment and the experimental treatment. And so very often, those standard genes or the control genes tend to be what scientists also refer to as housekeeping genes. They're genes that for all intents and purposes, we think are produced, transcribed, the same amount regardless of whatever else is going on in the organism. So very often these tend to be metabolism related genes. And in this case, let's assume that we're talking about a gene called G3PDH, which stands for glyceraldehyde 3-phosphate dehydrogenase. It's a housekeeping gene. We don't expect that its treatment will change from condition to condition. So that will be the red gene. And in contrast, the blue gene is the gene that we think, or we predict, or we hypothesize that the experimental condition will, in this case, increase its transcription rate. In other words, we would expect that in the experimental condition, there would be more blue transcripts than in the control condition, which is what's shown here in this figure. So again, the prediction is that treatment, the experimental treatment, will increase the transcription of the blue gene, which will result in our observing more blue transcripts. That is, more blue ESTs, expressed sequence tags. And that when we perform quantitative RT-PCR, we will see in the experimental condition more blue messenger RNA than we would have in the control condition. So again, that's the concept of RGE, or relative gene expression, is that we need to compare the amount of blue transcript to some sort of baseline standard that we don't think is going to be influenced specifically by the difference in the condition when we make whatever change it is from the control to the experimental condition. So if we just looked at these different experiments, and we've got 3 different scenarios here in rows. So the first one we looked at, if we just did interpretation of these data, comparing the control situation and the experimental condition, without normalizing the expression of blue to the expression of the red gene, which again we expect to be the same regardless of the treatment, let's take a look at what we would plot. So in the left side, in A, we have 2 blue transcripts, which we could detect by RT-PCR. And on the right side, we have 4 blue transcripts. So in column A, we've got 2 blue transcripts. And in column B, after treatment, we've got 4. That suggests an increase in blue gene expression, which is what the hypothesis was. So this sort of data by itself would indicate support of that hypothesis, that there's a correlation between the experimental treatment and an increase in the number of blue transcripts. Pretty straightforward. If we look at this second scenario, between the control group and the experimental group, we see 2 transcripts of the blue messenger RNA in column A, but after experimental treatment, we see only a single transcript. So that would be indication that there's a decrease in expression of the blue gene given the treatment. And that would suggest initially, although don't be fooled, this is the first step. Now we're going to look in a little bit at what happens when we normalize the data to the red gene expression. This data by itself suggests that experimental treatment causes decrease in expression of the blue gene. And that would cause us to reject our hypothesis. And then in this third example, you've got the situation where there are 2 blue transcripts on the left and on the right. And that suggests no change in gene expression, no increase or decrease of the number of blue transcripts. So that again would cause us to reject our hypothesis, because the hypothesis is that this experimental treatment should increase the amount of blue gene expression. So hopefully, take a minute, hopefully you see what's erroneous about these interpretations of the data on the left. Because some of these cells have less transcript overall, not just of the blue gene, but of the red gene also. And so the way I've drawn it, it seems to be like some cells have less transcription than other cells. That actually could be the case. What's more likely the case in these sorts of RT-PCR experiments is that when in the laboratory, scientists are collecting the tissues or cells from organisms in different treatments, they might accidentally get more cells in one sample than in another sample. And if you collect more cells, then you collect more messenger RNA. So it's not just the messenger RNA numbers might differ between individual cells, but it might be that the sample that an individual scientist collects, say, in a microfuge tube, might accidentally contain more messenger RNA total because they've collected more cells from one organism than from another organism. So the key point here is scientists cannot and should not make direct comparisons between independent samples, because there are all sorts of experimental variables that occur when you talk about different samples collected maybe at different times or from different organisms in different treatments. So what we need is a way to get relative abundance of the blue, not absolute. So what we've looked at on the left side were absolute number. We were just counting the absolute number of blue in the control condition and the experimental condition. This is where the red gene comes in. Because we predict or hypothesize that its abundance should be independent of whatever treatment we're using that differs between the control and the experimental condition, we can use the presence of and the number of transcripts of the red gene as a baseline. So for example, upper left, the top, we've got 2 blue transcripts and we've got 4 red transcripts. So if we normalize the number of blue to the number of red, if we take that ratio, which is what I'm indicating here, number of blue messenger RNA divided by the abundance of the red transcripts, and we plot that, then we see that there's 0.5 relative expression of blue to red. And if we take a look at this other cell, it's got 4 blue transcripts and 1, 2, 3, 4, 5, 6, 7, 8 red transcripts. Then that ratio, the relative abundance of blue to red, hasn't changed. It's still 0.5. So in other words, this may be that type of condition where a scientist collected half as many cells from a sample, thus half as many absolute number of messenger RNA molecules in one sample relative to another sample. So it's not valid to count the absolute number of blue messenger RNA transcripts in one sample and compare that directly to the absolute number of blue transcripts from the next sample. They need to be normalized to some control condition or some housekeeping gene expression in this case. So let's take a look at the other 2 situations. In the middle here, we've got 2 blue transcripts and 4 red transcripts at the start. And although initially it looks like the number of blue transcripts decreases from 2 to 1, so do the number of red transcripts. And in this case, I should have probably drawn a second one to indicate that both of those, again, were no change. So even though the absolute number of blue transcripts goes down, which is what the hypothesis is focused on, transcription is also reduced for other genes. So that doesn't mean that there's a specific effect of this treatment specifically on the blue gene. It means that overall there was some change in the experimental condition that caused transcription to decrease overall. Now remember, a minute or 2 ago, we looked at the number of blue transcripts here and the number of blue transcripts here, and we said, okay, there's no change in transcription of blue despite our hypothesis that this experimental condition should have more transcription of blue. But of course, the way I've designed this, we have 2 transcripts of blue on the left side and on the right side, but the denominator differs because here we've collected maybe a little bit more messenger RNA on the left side. So there are 4 transcripts, 2 red transcripts per blue, or 1/2 frequency. But what happens on the right side, we've got 3 red transcripts relative to 2 blue transcripts. So that actually shows a slight increase from 1/2 to 2/3. And that actually supports our hypothesis. So in sum, The normalization of gene expression from an experimental gene to a control or a standard or a housekeeping gene lets us avoid making false positives, seeing that the absolute number of blue increases when it really hasn't, giving us the impression that our hypothesis is true when it's not. Or looking at the absolute number of blue decreasing in the experimental condition, giving us a sense of a false negative, when actually the relative abundance of blue to red has increased in this experiment. And then here we see that even though there are 2 blue transcripts on the left side and 2 on the right side, the fact that the blue relative to red are more abundant on the right does support the hypothesis and shows an effect that this experimental treatment does actually increase the expression of blue relative to other genes inside the cell. Now to conclude, let's look at the last concept that's essential for right now, and that's the concept of epistasis. And you may have seen this before in papers like this one from Trish Wipkoff, looking at the effects of these different proteins. So this is a review figure indicating that enzymes like tyrosine hydroxylase convert tyrosine to DOPA, and additional enzymes like yellow convert DOPA into a black pigment. And if those pigment precursors continue down this pathway, they can produce brown pigment using an unknown enzyme to convert dopamine to that dopamine melanin, or then the ebony enzyme to convert dopamine to NBAD and eventually to NBAD sclerotin, which is a yellow pigment. The idea of epistasis and the question behind epistasis is trying to figure out which order different enzymes act in a pathway to produce particular phenotypic outcomes, like the different pigment colors. So without preliminary information, the way that developmental biologists proceed with figuring out what order different genes and their proteins that they encode act in a particular pathway is a particular type of set of experiments where you look at sets of mutants. That should be tyrosine hydroxylase, and we'll also look at ebony. So let's recapitulate how geneticists figured out which order of proteins act in this pathway to produce different pigments in fruit flies. And the way this works is we look at, first, let's look at the wild type organism where they have the tyrosine hydroxylase enzyme and they have the ebony enzyme. And we're going to look at what is the trait that's produced with that combination of proteins present. So again, tyrosine is converted by tyrosine hydroxylase to the precursors for all of these different pigment types. And the production of yellow pigment depends on the presence of ebony. So when tyrosine hydroxylase, the gene, is present and produces the protein tyrosine hydroxylase, and the same for the ebony gene and the protein it produces, then the trait is the organism can produce black pigments, brown pigments, and yellow pigments. But that doesn't tell us anything, although we can see the drawing here of this pathway, it doesn't tell us yet whether or not ebony might act upstream of tyrosine hydroxylase or vice versa. It doesn't tell us the relative order of when those different genes need to be present to allow the downstream genes to do their jobs to produce these different pigments. So then, the epistasis experiments involve using combinations of different mutants, both the single mutants, where they're lacking one or the other of the genes, and then comparing that against the double mutant that lacks both of those genes and proteins. And we're going to look at the different traits that are produced. So what will happen if we have tyrosine hydroxylase present, but no ebony? So tyrosine hydroxylase is present. It will convert tyrosine to DOPA, so we can get black pigmentation produced. And then DDC is present, so DOPA can be converted to dopamine and then through an unknown enzyme to brown pigment. But the question is, can we get yellow pigment produced? Without the ebony protein, we never get yellow protein. So that's it. In the TH positive but ebony negative, ebony mutant flies, we would see black pigment and brown pigment produced, but no yellow. Now, how about the opposite single mutant, where tyrosine hydroxylase here is absent? If there's no tyrosine hydroxylase, then tyrosine never gets converted into DOPA, and DOPA is a precursor to all of these different pigments. So if there's no TH, there's no DOPA, there's no black pigment, no brown pigment, no yellow pigment. So in that single mutant, we get no pigment production. Now, to figure out which gene, tyrosine hydroxylase or ebony, acts first in this pathway, we now need to look at the double mutant. What is the trait when neither tyrosine hydroxylase nor ebony is present? And we just discussed that when tyrosine hydroxylase is absent, no pigments get produced. This is the important point, regardless of whether ebony is present. And that's the key here, is that the downstream gene, and that's the concept of epistasis, a gene that acts downstream of another gene depends entirely on the presence of that upstream gene to do its job in producing some particular phenotype. So here's how these sorts of experiments are analyzed. Once you look at the single mutants and the double mutants, and you look at the traits that are produced, you look for the common trait. In this case, there are two different conditions where no pigments are produced. Then you look for the correlation with the mutation. Which gene was mutant every time no pigment was able to be produced? That was mutations in tyrosine hydroxylase. So regardless of whether or not ebony was present or absent, the absence of tyrosine hydroxylase allowed the production of no pigment. And that is what defines tyrosine hydroxylase as acting upstream, because the downstream protein, ebony, its function, its role, its ability to produce yellow pigment instead of brown pigment depends on, is epistatic to, the presence of the upstream gene. So that's the concept of epistasis. We want to know what the wild type trait is. We want to look at the single mutants and then the double mutant. And whichever of the single mutants has the same phenotype as the double mutant, that is the gene that acts first in the pathway of whatever process is being looked at. Related again to hedgehog and Wnt signaling, I want to remind everybody that when we look at RNAi phenotypes of hedgehog or patched or cubitus interruptus, which is also called GLE, and when we look at the knockout phenotypes or the RNAi phenotypes of beta-catenin, remember that when we get rid of those proteins by RNAi or any other means, the phenotype that's produced is going to be the opposite of the wild type or normal role of that protein. And so I'd like you, if you want to use this worksheet to write out what is seen in this paper when these different proteins, genes, are knocked down. And therefore, based on that, what is the normal function of those different proteins? And then predict or report the effect of RNAi of those genes, how each of those would change Wnt expression. So again, we have two different pathways here. We have the hedgehog pathway, and the Wnt pathway. And we're going to see whether or not these two pathways interact with each other, and that's what this column is all about. So I hope you'll take advantage of this spreadsheet to try to make sense and organize your thoughts and the findings of this paper so that we can discuss these during class and in our discussion boards.